Hello, Dr. McDonald here. Today we're going to discuss the various, I should say, because there are a lot of different cells and organs that make up the immune system. Once you get the hang of who is where and what they do, the immune system is really cool. And of course, it's something we're trying to manipulate all the time because it has so many applications to so many disease processes. So instead of my going on and rambling about how wonderful I think the immune system is, let's learn about it. As I said, the immune system consists of many different organs, which you can see with the picture over on the right. The primary lymphoid organs, where the cells are developed, are the bone marrow and the thymus. The secondary lymphoid organs is where the cells of the immune system interact with infectious microorganisms or foreign proteins are in the secondary lymphoid organs, and those are in the lymph nodes, the spleen, and of course, mucosal associated lymphoid tissue. So basically all the lymph nodes will protect for the tissues, the spleen, anything that gets in the blood, and the mucosa, anything that gets into the respiratory tract, genital urinary tract, all that good stuff. We have our troops ready and waiting to fight infection. Unfortunately, there are a lot of cells in the immune system. And there are two parts to the immune system. There's what we call the adaptive immune system. And the cells that mediate the adaptive immune system are white blood cells, otherwise known as leukocytes. And they're known as lymphocytes, as you see over here on the blood smear to your right. But the innate immune system has a whole bunch more. And these leukocytes, or white blood cells, are known as myelocytes, and they include neutrophils, monocytes in the bloodstream, macrophages in the tissues, same cell, dendritic cells, basophils. They sort of think basophils and mast cells are related. Basophils are found in the bloodstream, mast cells in the tissues, and eosinophils mostly in the tissues, very little in the bloodstream, but very low numbers. And the interesting thing is that all of these cells derive from a single hematopoietic stem cell. And then it'll break off into either the lymphoid or the myeloid branches of the white blood cells. Upon cell division, a multipotent hematopoietic stem cell, that's what HSC stands for again, has two possible fates. It can regenerate itself, otherwise known as self-renewal, and it's forming another hematopoietic stem cell. Or it can differentiate into one of the possible progenitor cells that I mentioned on the previous slide. It could be a lymphoid progenitor cell, in which case, guess what, guys? There's more than one type of lymphocyte, where it can then become a T cell, a B cell, and a natural killer cell. Or it becomes a myeloid progenitor, in which case it becomes everything else: monocyte, neutrophils, eosinophils, basophils, platelets, and even red blood cells. Now, the progenitor cells have lost the capacity for self-renewal, and so they are committed to forming one of those cells. And commitment, of course, is to a particular cell lineage, depends on what receptors they are expressing and what growth factors and cytokines are present in the environment or have been sent to the bone marrow, signaling they need more than one particular type of cell. Now, aren't you glad you don't have to sit here and learn all these different cells and be able to identify them, which a pathologist would do, or a clinical laboratory hematologist, which I once did for a little while. Anyway, the mature cells aren't so bad. The committed progenitors, they're all blast cells and they all look alike. But anyway, this just gives you an idea of how it starts with a self-renewing stem cell, and then it's either a common lymphoid progenitor or a common myeloid progenitor, and then you get all your different cells. Just wanted you to see how they're derived. That's all. Don't worry, you're not going to be tested on this. However, you will be tested on all the different cell types. So here we go. First one we're going to talk about is monocytes and macrophages. These are, of course, derived from the myeloid lymphoid cells. They arise in the bone marrow. They leave the bone marrow as what we call a promonocyte. Don't worry about this so much. But then they differentiate into a mature monocyte actually in the blood. They circulate in the blood for only about 8 hours, during which they get a little bit bigger. And then they'll migrate into the tissue and become a specific tissue macrophage, or in some cases, a dendritic cell. Now, originally we didn't realize that all these different cells, such as histiocytes and Kupffer cells, were all macrophages. So they looked different from each other, so we gave them names. And you can see all the names that the different macrophages have in the different tissues. Microglial cells are macrophages. But if you look at the monocyte that's in the blood, you see over here on the lower right what they look like. They have a very large nucleus and some cytoplasm, a lot more than a lymphocyte. And their nucleus actually looks like there are lots of things going on in there. And that's what is the case for monocytes, and it's definitely for macrophages. So this picture doesn't really show you that much difference, but it does show you one of the things that it's noted for the macrophages is the fact that it is phagocytic. And so it will bring in microorganisms in a phagosome, which fuses with a lysosome to form a phagolysosome so they can kill those nasty bugs. As I was saying, macrophages are bigger than monocytes because they have an increase in the organelles, and they're better at phagocytosis. So they're more capable of ingesting more microorganisms. They also have an increase in the amount of the enzymes in their lysosomes that will help to break down the bacteria and maybe some extracellular viruses, and they're fixed within the tissue. Sometimes they can move a little bit with some amoeboid movement, but not so much. What we'll find out is if, however, it's a dendritic cell, yes, it can be very motile. Now, macrophages are activated by a number of things. If they phagocytize a particular antigen, that activates them. T cell cytokines, they have receptors for a number of cytokines produced by T lymphocytes. They get activated that way, or something that we've already learned, pathogen recognition receptors that they have bind to pathogen-associated molecular patterns, so the PERS, the receptors on the macrophage bind the PAMPs on the microorganism, and that's how, in many cases, they are able to phagocytize them. Once a macrophage is activated, it has three major functions, and this is the sort of thing I do like to ask. They have enhanced phagocytosis, so as I've already said in the previous slides, they increase the killing of any pathogens that they ingested. They're going to increase their secretion of cytokines and cytotoxic proteins, okay? Again, hoping to kill off microorganisms. And they'll also have something that's very important. They have an increased antigen. That's what the AG stands for, antigen. I apologize for using the abbreviation here without explaining it. They have increased antigen presentation to T cells because they've increased expression of class 2 MHC and other things that we'll learn about known as co-stimulatory membrane molecules. So if you look at this picture right here and follow my cursor, here's the T helper cell, and here's the antigen presenting cell. So think of this as the macrophage, and it's got the class 2 MHC on it, and it is presenting a portion of a microorganism to the T cell receptor. And T cell receptor for antigen, as we learned about, and then it has this other receptor that can bind to the MHC, and this helps to activate the T helper cell to get the T helper cell to start secreting cytokines and everything. Granulocytic cells are all part of the innate immune system, and we're just going to go over their functions briefly here. Neutrophils are phagocytic. They're the first to arrive at a site of injury where there might be microorganisms. Eosinophils are motile, but they have limited phagocytic ability. Basically, they're really important for secreting compounds that will help fight against infection with parasites. And they also have receptors for IgE, which means they can mediate or participate in allergy and asthma. Now, basophils also release active substances during allergic responses because, again, they also have receptors for IgE antibodies. And, again, they're also important in parasitic infections. And then there are the mast cells. They believe the precursors are in the blood, and then they get into the tissues where they differentiate into a mast cell. And they line just outside all of the blood vessels, particularly the smaller capillaries. And what they do is, if there's an injury to a blood vessel, they release all these pharmacologically active substances. And yes, they can do this during allergic responses, but they help to also initiate inflammation for any kind of reason, not just allergy. So they're very important in the initiation of inflammation. This slide just shows you a number of the cells, kind of what they would look like. Not really well over here on the left, but on the right, this is a granulocyte. You can see all the, this is an eosinophil, I should say, because it has the orange, sort of pinkish, dark pinkish granules. This is a neutrophil because it stains with neutral dyes, and basophil stains with a basic dye. And this is, all the little dots are the granules and why they're called granulocytes. Dendritic cells are very important. They got their name because they have long membrane extensions, so they look like neurons, okay? So they have the dendrites. There are many different types of dendritic cells, but they all have one major function, and that is to present antigen to T helper cells. In fact, they're the best at it. They're the best antigen presenting cell, or APC, for what we call naive T cells. These are T cells that have a T cell receptor, but they haven't yet encountered their antigen. Now, the four types of dendritic cells we'll mention here, they're the Langerhans cells that are in the skin. Then we have interstitial dendritic cells, which are located within various tissues. Then you have the monocyte-derived dendritic cells. They migrate through tissues, lymph, and blood. And these plasmacytoid dendritic cells, they're kind of weird. They're part of the innate antigen-presenting cells, but they all express very high levels of class 2 MHC and co-stimulatory molecules that help activate the T helper cells. Now, there's one last type of dendritic cell. It's a follicular dendritic cell. They don't develop in the bone marrow, so they're not coming from the hematopoietic stem cell. And they don't present antigen, but they bind antigen antibody complexes, and they interact with B cells in follicles like lymph nodes and stuff. So they're very important in generating an antibody response. So you can see here, just kind of simplified again, just so you have an idea where they're coming from. Here's the hematopoietic stem cell, here's the common myeloid progenitor, and then we get all these different types of, and notice that we don't have the follicular dendritic cell here, we get all the different types of dendritic cells. So we finally can talk about lymphocytes, which mediate what we refer to as the adaptive immune response. They do indeed mediate adaptive immunity, and I forgot I had put it in there like that. I promise never to have it move quite like a typewriter again. Anyway, we have lymphocytes are up to 40% of our white blood cells. We have a lot of them in the adult. And they are constantly circulating in our bloodstream, in our lymph, because they are our troops that are basically marching the perimeter, making sure that there haven't been any pathogens that have gotten into our body. Now, there are three major populations. They're the B cells. They mediate what we call humoral immunity, which we talked about in the previous lecture. And they do that because they produce antibodies which are present in the humors of the fluids of the human body. T cells mediate cell-mediated immunity, and they're going to produce cytokines. And unfortunately, there are a ton of different types of T cells, as you'll find out. And then we have natural killer cells. These are large granular lymphocytes. They don't have B and T cell markers. In other words, they don't have a T cell receptor or B cell receptor. But we'll find out a little bit more. And they're actually, the natural killer cells, not part of the adaptive. It's part of the innate immune system. The first type of lymphocyte we'll talk about are B lymphocytes. As you already know, they arise and mature in the bone marrow of men. But they were originally discovered in the bursa fabricis, which is an organ that we don't have in chickens, of all things. But fortunately, bone marrow also starts with a B, so it's easy to remember. Anyway, mature naive B cells will have antibody on their surface. That is referred to as a membrane-bound immunoglobulin. And the thing is that it is the B cell receptor for antigen. So that surface antibody, as it says, serves as the receptor for antigen. It binds antigen directly, which is very important. It binds soluble antigen or antigen bound to a cell or a microorganism. Because usually what it's actually binding is a very small part of the pathogen. And the antibodies can also be secreted, so they are in the fluids of the body. Now, what do you need to do to activate a B cell? Well, it has to bind antigen on its membrane-bound immunoglobulin or antibody. It has to interact with a T cell, and that T cell is going to stimulate it by secreting particular cytokines. And then you have an activated B lymphocyte. What happens when the B lymphocyte is activated? Well, it proliferates and it differentiates over about 4 to 5 days and forms a population of either effector plasma cells. Plasma cells secrete antibody. That's their major function. And or memory cells. Now, the memory cells are long-lived. They have lots of antibody on their surface, and they are ready for the pathogen that stimulated them and ready to eliminate before they cause any injury, so they won't cause disease. The plasma cells that secreted a ton of the antibody, they die within a period of time, and then but you have the memory cells to protect you. Okay? And I thought I'd show you what a plasma cell looks like. I wanted you to notice that lymphocytes usually just have a huge nucleus and very, very little cytoplasm. And what you're seeing here with the plasma cells is a huge amount of cytoplasm staining this really pretty blue. And that's indicating that all it's doing is producing antibody molecules, tons of them. A single cell that you have to use a microscope to see will produce a gram of these a day. It's utterly remarkable. And then there are the T lymphocytes. They arise in the bone marrow, but they leave very early and then migrate to the thymus where they mature. Now, the receptor for antigen on a T cell is again called the T cell receptor, and it's different from an antibody in many ways. First of all, it does not bind antigen directly. It can only bind small bits of an antigen that are bound to a major histocompatibility protein, or an MHC. Now, the T cell receptor is actually a complex of proteins. Again, a protein is also known as a polypeptide. It's the same thing, which includes what we call this complex CD3. All right, now here's where T cells get a lot more complicated than B cells. There are functional subsets of T cells. Okay, T helper cells express CD4, and they will bind to antigen with their T cell receptor on class 2 MHC and secrete cytokines. And yes, there are subsets of T helper cells. For our purposes, we'll just stick with T helper 1 and T helper 2. TC, or T cytotoxic cells, are also known as cytotoxic T lymphocytes. And I think I'll put that at the end to remind you. T cytotoxic cells express CD8, and their T cell receptor will bind to antigen on class 1 MHC. And then they will actually end up killing the cell presenting the antigen. Now, there are what we call T regulatory cells, and they express CD4 and CD25. They like CD4 because they have actually CD4. They will bind antigen on class 2, and they suppress the immune response. Remember, we have to get back to homeostasis. Once the immune response is activated, it's got to be shut down once the pathogen is eliminated. So all three of these, as it turns out, can be progenitors of T memory cells. It is important to keep straight which CD markers is on which subset of T cells. Sorry, but it is. The CD4 is exclusively found on T helpers and Tregs, okay? And it makes the T helper cells recognize antigen on class 2 MHC. So the fancy immunological term is it restricts T helper cells. So it dictates the fact that T helper cells will interact with antigen on class 2 as opposed to class 1. So the CD4 does that because it itself binds to CD4, whereas CD8 is almost exclusively found on cytotoxic T lymphocytes, otherwise known as T cytotoxic. And they restrict the CDLs to antigen recognition of antigen on class 1 MHC, so not surprisingly, it binds to class 1 molecules themselves. Now, the problem with lymphocytes is you can't tell one from the other. You can't tell a B cell from a T cell if they're naive T cells and they haven't been activated. And even when they've been activated, you can't tell a B from a T or a CD4 from a CD8. So what do? Well, there are cell surface molecules. These are cell surface proteins. They can also be antigens. And they're found on the surface, of course, because they're hooked into the plasma membrane. And they are recognized by antibodies that we've injected in different animals so that we can identify the different cell types. So all of the monoclonal antibodies that react with a particular surface antigen are grouped together and they're called a cluster of differentiation, or CD. You know what I call it? Cell differentiation. So you can differentiate one cell from another. There's more than 200 of these, so don't worry about it. But you do need to know a few of them, CD4 and CD8. This table is just one of many that you can find. In fact, this is a short table of all the CD molecules that you can see. Some of the more common ones that you might hear are CD2, 3, 4, 5, 8, and 16 and 21 and 28 are actually ones that we will talk about at some point in the next few weeks. So the rest, however, I wouldn't worry that much about. But if you have a little idea that they are membrane proteins and we use them to differentiate one cell type from another, it doesn't make it seem quite so scary. And then there are the natural killer, or NK cells. They are lymphocytes, but they're larger and they have granules in them, which makes them cytotoxic against tumors. In fact, that's why they got their name. They were found to be naturally cytotoxic against tumor cells without any prior exposure. We later found out, though, that they actually make up to 10% of all our lymphocytes, and they are also cytotoxic against virus-infected cells, but they do not have a T cell receptor for antigen. So how do they recognize that a tumor cell or an infected cell is somehow abnormal if they're not recognizing a specific antigen on class 1? Well, one of the things they do is actually recognize reduced expression of class 1 on tumor cells and other tumor-associated antigens, and on viruses too. Some viruses decrease class 1 expression, so cytotoxic cells aren't activated, but we've got NK cells that recognize this. They also have a receptor known as CD16, which binds the stem portions of antibody molecules, and that enables them to destroy the target that way. So they are important in a process that we call antibody-dependent cell-mediated cytotoxicity, or ADCC for short. And we'll talk about that a little bit more later on. Now, I wanted to give you a little bit of a clinical reference to some of these things you're learning here. And it turns out that NK cells are very important against some tumors, as demonstrated by this disorder. This is a syndrome that can be inherited called Chidiac-Higashi. It's autosomal recessive, so here we are back to genetics. And the neutrophils and macrophages and NK cells are impaired in these people. And as a result of this, they have a very high incidence of lymphomas. We learned more about it as we found out that there were mice with a beige mutation that lack NK cells, and turns out that they grow injected tumors better than others. So you can see that NK cells are very important. So I couldn't resist putting a little picture of a beige mouse. They are kind of cute. And then, of course, here is a large granular lymphocyte. You can see that it does have some granules in it right here, and of course it's larger and has a lot more cytoplasm. Finally, we have something called NK1 T cells, or NKT cells. NK1 is actually an old version of it. They have characteristics of NK cells and T cells. Remember, NK cells and T cells are both lymphocytes. They have a T cell receptor, unlike NK cells, but they are not as variable as the T cell receptors on regular T cells. They're called semi-invariant. So frequently we refer to them as invariant NKT cells with a little I in front, as you can see down here on the bottom of the slide. And what they do is they enable these cells to recognize a lipid as opposed to a protein antigen on a CD1 molecule that's going to be on the cell that's much like an MHC-like molecule. So these NK1 or NKT cells also express CD16 and can kill cells. And when activated, they secrete a ton of cytokines, which will promote inflammation and antibody production. And I don't want you to worry about this particular sort of flowchart over here. I just want you to see that they can interact with a lot of other different types of lymphocytes, T cells, Tregs, B cells. And so the NKT cells, or NK1 T cells, are part of a rapid response that provides early help while the adaptive response develops. So now we can actually go in and talk about the different organs. We'll start with the primary lymphoid organs and begin with the bone marrow. It's the site of B cell development and maturation. B cells also undergo a selection process. Remember, we don't want the immune system to react to self. We want it to be tolerant of self. So while it's maturing, there's a process that will eliminate B cells that rearrange and get a B cell receptor that binds self antigens. So those self-reactive antibodies potentially are eliminated. Now, the hematopoietic stem cells, of course, are going to be in the bone marrow, and they grow and mature on what is called stromal cells. Stromal cells are not going to participate in becoming a white blood cell, but they do help support the growth of all the different white blood cells, myeloid and lymphoid, and of course push the hematopoietic stem cell to one differentiation or another. And these stromal cells include fat cells, fibroblasts, endothelial cells, even macrophages. And what these cells will do then is provide an environment for developing all the white blood cells that make up, again, part of the immune system. So they also produce an extracellular matrix, and they secrete growth factors, which help for the development of these different cells. So it's a very important, what we call cell generating organ. And I couldn't resist putting a couple of things in here. This little picture here on the left shows you, of course, the bone marrow and all the different things that it can influence, etc., and the cells that will get developed. And then, of course, another little picture of a long bone here. And then this is what a typical bone marrow slide looks like. And you can see all these developing cells. The very large cells are blast cells, and they're not adult yet. The other primary lymphoid organ is, of course, the thymus. It is where T cells develop and mature. It has two lobes. It's just above the heart, and it's surrounded by a capsule, and it has connective tissue separating it. I don't care if you remember all that. It just gives you an idea that it's separated into areas, and what will happen is different things occur in these different areas within the thymus. So you've got an outer compartment, that's the cortex, and you have a lot of immature T cells in there, and then the inner part is the medulla, and it has fewer cells, but they are mature T cells. And when they're in, T cells are in the thymus, they're referred to as thymocytes. So if ever you hear that word, that's what they're talking about. It's T cells. And then they also have a stromal cell network like the bone marrow, but it's not as extensive. And within that network, though, we have epithelial cells. Dendritic cells are very important, and macrophages. And we'll find out all of their roles later on. This, of course, is a cross-section of thymus, showing you how it's separated. You've got the outer, which is the cortex, and the medulla in here, where they are older cells and everything. I'm not going to obviously test you on it, but I thought it would be nice for you to see a picture. So what is the function of the thymus? And that's probably more important than knowing that it's got trabeculae and all that good stuff. It produces T cells that are going to protect our body from infection. During the time the T cells are developing in there, they will develop what we call a repertoire of T cells. And this is, so we have a wide variety of T cells with different antigenic specificities. In other words, the different individual T cells will have a different T cell receptor that arose from these random gene rearrangements, which we'll talk about later, getting a unique T cell receptor protein on its surface. And while these rearrangements are occurring, they're checking to make sure that that T cell receptor will, if it reacts with self-antigen, it's eliminated, because that would induce autoimmune disease. Now the weird thing is that the thymus shrinks as we get older. Yeah, it reaches its maximal size at puberty, and then it atrophies. But it still helps provide us with T cells. And that takes us to a discussion of the secondary lymphoid organs, which includes the lymph node, spleen, and all the different mucosal-associated lymphoid tissues, the adenoids, tonsils, Peyer's patches, and appendix. What's important for you to understand is all in these secondary lymphoid organs, they are populated by mature B cells and T cells that have come from the bone marrow and the thymus, respectively. They have receptors for antigen, and they have not encountered antigen. They're circulating around our body to provide surveillance to see if an antigen to which they can respond has entered. And so what we find in there, in the lymph nodes and the spleen and everything, is all these cells that are capable of responding and eliminating infection regardless of where that infection occurs in our body. Now, the lymphatic system is very important in protecting our tissues from infection. It's a system of vessels, of course, that it's going to help to drain excess fluid that gets into the tissues and return it back to the bloodstream. The fluid within there is referred to as lymph, and it's pumped by action of muscles and everything. I know as nurses you guys know this pretty well. But what happens is the lymph will trap or catch any pathogens, microbes, or this sort of thing, and then drain them into the lymph nodes. And in the lymph nodes, we have all the B cells and T cells and macrophages and dendritic cells waiting to see if the pathogen to which they're specific enters, and if they do, they get activated and they're going to eliminate them. The lymph nodes are sort of these strange looking organs. They're bean-shaped and they're packed with lymphocytes, B and T, and macrophages and dendritic cells, just waiting to launch an attack should a pathogen get in there. And they're also strategically placed at the junctions of lymphatic vessels. So anything that gets into the tissue will drain and be delivered to the lymph node. Okay, LN stands for lymph node. Well, so the lymph is percolating and carrying the antigen. It's trapped by the phagocytic cells and the dendritic cells. And then, of course, things can get presented to T cells and activate B cells. So the lymph nodes are actually divided into three regions. There's the outer region, outer cortex, is T independent, so it's going to have lots of B cells and macrophages and what they call follicles where dendritic cells are, and this is where they're going to activate B cells to make plasma cells and secrete antibody. Now the middle part is where the T cells are, so this is the T dependent area, and they've got lots of those dendritic cells which are super good at activating T cells, and they're there to activate the T cells. And the final inner area is very sparsely populated with lymphoid cells, and many of these have plasma cells that are actively secreting. Because once they get into the inner, they're usually effector cells, they've been activated, and they're going out to get rid of that pathogen that got into the tissue. It would be nice to just take a quick look at what a lymph node might look like. You can see over here on the upper right that they're kind of weird looking. And then if you look down at the picture so that you get an idea how it's separated, you can see that there are areas, germinal centers, these are where the B cells are, and then we get into the other areas, we would see more of the T cells and things like that. So and of course the lymphs are bringing in the fluid, which is trapping the antigens, and everything is there to protect us from a pathogen that enters into our tissues. Then there's the spleen. Now the spleen's function is to mount an immune response if an antigen, or a pathogen if you will, use antigen, is in the blood. It also recycles old and damaged red blood cells and platelets and things like that. So it specializes really in filtering blood and trapping any blood-borne antigens, think of pathogens, or systemic infections. It too has a capsule around it like the lymph nodes and has tuberculated formed inner compartments. You have the red pulp, which is a storage site for red blood cells, and this, I apologize for this old abbreviation that I used here. This is for macrophage. That's a Greek thingy. So every now and then I forget about that, and I put in one of my old abbreviations. Anyway, so macrophages are there to help remove the old and defective red blood cells. Then you just have what they call a marginal zone, which separates the red and the white pulp, and it contains a lot of macrophages and certain types of B lymphocytes that are unique. And then the white pulp, which surrounds the splenic artery and forms, of course, with a lot of T cells, a periarteriolar lymphoid sheath, which of course we call PALS for short. And this is where you have lymphoid follicles with B cells and T cells in it. And this slide again is just showing you very briefly the normal spleen, a kind of general picture of it, and then where you would have different cells and follicles and everything. And of course this where is the red pulp, and then we have the white pulp, and etc. So again, I'm not going to test you on the anatomy, but it's just kind of nice to see a picture of these here and there. That leaves us with a discussion of the mucosal associated lymphoid tissue, or MALT for short. We're talking about the mucous membranes, the respiratory tract, gastrointestinal, genital urinary tracts, and they're all defended by lymphoid tissues. They're kind of organized, but not nearly as much as lymph nodes and spleens and stuff. And if they're part of the pulmonary system, they call them bronchus associated, so it's BALT, or the gut, it's GALT, you know. They like, immunologists really like abbreviations. So Peyer's patches, which are PP, these are lined up intestine, tonsils of the respiratory tract. These are all part of the mucosal associated lymphoid tissues. And they're going to defend if organisms, pathogens come through any of those routes. The Peyer's patches through the GI system, the tonsils through the nose and respiratory tract. The malts, again, will also have plasma cells in them, and usually a lot more than in the bone marrow, and then the lymph node and spleen combined, because this is an area where if you have an antibody that can bind to a structure on the pathogen that helps them attach to our tissues and stuff, you can prevent an infection entirely. So having antibodies there is really useful. They also have specialized populations of lymphocytes and epithelial cells, again, that they help to protect the GI tract. In fact, they have things that they call M cells. And these are just epithelial cells in the mucous membranes, but they can transport an antigen from one side to the other, okay? And that helps to activate T cells and B cells and then lead to production of the antibodies that can prevent them from causing an infection later on. And you can kind of get that feeling here, where here's the antigen and it gets through and it's coming through it, and so it can be presented to the macrophage, and the macrophage, I've put a couple of different pictures up there so you can get an idea of how it works. And then we can even talk about it in the skin. The epidermis does have keratinocytes, which are specialized epithelial cells, and they form our impenetrable barrier known as the skin. They also secrete a lot of cytokines that can induce a local inflammatory response. Sometimes they can even function a little bit as an antigen presenting cell. But what's really important are the Langerhans, or dendritic cells, that are present throughout it. And they're very phagocytic, and when they do that and internalize the antigen, they then leave the skin and move to the nearest lymph node. Okay, think of it as a fort with all the troops waiting, where they can differentiate into these other kind of types of dendritic cells and activate the naive T helper cells. So the underlying dermis of our skin contains a lot of T helpers and T cytotoxic and memory cells and macrophages, and they are actually this is why when you give a vaccine, one of the best places to generate a complete immune response is in the skin, because of the presence of these Langerhans dendritic cells. So that's it for just learning what I call the cast of characters in the immune system, the cells and the tissues, and we'll move on to the innate system in our next lecture. Goodbye.